Bismillah ar-Rahman ar-Rahim. We are about to start our second session of the morning. Please return to your seats. Assalamu alaikum and hello everyone. I am Dr. Shaukat Khan, Assistant Professor at Chemical Engineering Department, Dhofar University. I will be chairing this session. We will have two presentations in this session. Is Dr. Muhammad already presented in the first session. Our first speaker is Dr. William Zimmerle. He is affiliated with New York University, Abu Dhabi, UAE, and Farley Dickinson University, USA. And the topic of his presentation is sensing resinous material in the Southern Arabian Peninsula, annotating the data features of a chemical heritage library of the study of traditional Arabian scents. Please welcome Professor William.
Thank you. Can everyone hear me? Right. Perfect. Thank you very much. It's an honor to be here this afternoon. I thank Dr. Rashan for the invitation, and Dhofar University and the Board of Trustees and the organizers of the conference for hosting this conference today. For anyone from the GCC, and particularly for people from Dhofar, this image doesn't really need a defense or warrant for investigation because it's quite known to all of you. Most of you come from families, have family members who have made these who over the last, at least of the last decade. But how we look at this and what this can signify further for us to understand in terms of the trade routes that ran in Arabia from antiquity right up until the present day, the movement of resins is yet really to be interrogated until now. And so today I want to begin my discussion sort of with the principal component parts, just briefly, of the material culture side of things. So if you permit me to do a little archaeology, and that's how I'm trained, we'll look about 10 slides at the archaeological heritage things, material culture, and then move our attention over then to the problems that we have there and how we are trying to solve them from the chemistry data that we're accumulating as we're building a library of resins for the study, particularly of heritage and archaeology. And I'll add here, it's really a privilege to be back in Dhofar University. In 2011 to 2012, I was awarded funding from the Diwan of the Royal Court to do the ethnography of this work, which you'll see in a moment, the map that we have. And then from 2012 to 2013, I was a United States Fulbright scholar, senior scholar here at the university. So it's good to be back here. And then I even had so much enjoyment from being here that I came back 2 years later as a faculty member for a year teaching in the social sciences. So it's good to see so many faculty members here that I had met before. Incense burners themselves resonate with the Omani community, and this is one of the earliest one, as many of you know, that we have in the archaeological history. Found, excavated properly in the 1990s, up north from here on the coast of Ras El Jins. This particular form made of sandstone was found in a house, upside down, turned around, and the museum in Muscat allowed us years ago to do a 3D image of it. We can talk, as I said, about the principal component parts of the surrounding form. And what I want you to understand that making a square form is not an easy thing to do, whether it's hand mold or whether it's cut from stone. It requires quite skill, as many of you know from Dhofar. But today's lecture I want to talk about, because other lectures I've talked about the form a bit. Today's lecture I want to talk about the inside. What can we glean from the analysis of the residues that are found in incense burners themselves? It's reported that this was tested for Boswellia sacra, but we haven't been able to find the report. So this is something that in the future we would want to do retesting of to be sure of the analysis. You can see the excavated form where it was found, and archaeologically we have a growing number of incense burners formed, which are testaments to the Arabian trade that ran in many directions, starting from Salalah where the trees grew, the frankincense trees, and going in all sorts of directions north. And I'm proud to say we've done over the last decade quite an extensive ethnography survey of treating the Oman as a heritage park of incense-related material culture. So we've worked in every major part of the Sultanate now, documenting ethnographically the forms from storytelling to videography to photography, and we've reduced it in book form, which towards the end of the presentation you'll see. The maps start to look something like this from the archaeological side of things. This form, as I said, is difficult to make, but once the Arabian trade gets underway in the first millennium BC, we begin to find it in every quadrant, this form, in various evolutionary forms, in every quadrant of the Middle East. And here you just see a basic map, and you could split these sides from east to west. When we look at this kind of data, we can also match it up with the historical data that we have. And it raises a number of different linguistic, etymological difficulties for us as historians, archaeologists, heritage preservers. One is the terminology in the ancient languages is very not specific enough. So one term we will see again and again in the Neo-Assyrian annals of the 8th and 7th century is that what was taken from as a camel raid, as a dromedary raid, were spices or aromatics of all kinds, riku kalama. Very broad, right? We just don't know what kind of spices and aromatics or resins. And so this becomes our first problem, but we know that they're in circulation roughly by the 7th century BC. When the trade routes are running in every direction, everything in red you see here is basically resins moving in the north from Arabia, and everything in blue are resins moving in the south from Africa, all in the first millennium BC. We can map this further, and this is a simpler map in some ways, but it actually shows us some very good information. We can see two very specific different lines of evidence in terms of the trade routes. On the west side is the overland trade, moving up from where frankincense grows, the trees grow, further north, making their way up to Gaza. And the Roman historian Pliny the Elder tells us that if we were to use inflation today and calculate it, we're looking at roughly around 1 century BC, $35 to $40 per gram for frankincense because of the tolls that the frankincense has to pass through. It's getting taxed along the way, something we're all familiar with, taxation. It existed in all periods of time in the ancient world. On the other side, we see another route, right? And that's the water path route on the eastern side. And we know that there was movement there. And the reason we know this is we ask the fundamental question as archaeologists, how do we track a trade that's based in combustibles? And it's based on what's left behind. So small incense burners, on the west side they're made of stone, on the east side made of clay, are deposited all along the landscape, allowing us to sort of light up the map and to see where these routes ran. But the incense burners, from our analysis, are generally locally made, which shows us that what is really lucrative in the ancient world are the resins, not the burners themselves. But as the resins move forward, gum resins particularly, increase in value, you need something to burn, a particular technological device to burn the resins. And that's why we find so many of these forms every quadrant of the Middle East. For example, in Najran, across the border in Saudi Arabia, just a little rain, for example, will expose the legs of an incense burner right onto the mound of the site. We also have additional evidence epigraphically that's useful for us. One particular incense burner, about 6th century BC, has the name Lebona, frankincense, listed on it. We've never tested it, but it's inscribed on the side of the incense burner, further testifying the trade of frankincense. And we know that this word luban, that we keep using again and again in Arabic, is a descriptive term. It's one of the oldest Arabic words that we have in our etymological history. It exists as early as the 6th century BC, coming first in Greek poetry, the Greek poet Sappho, but it's an Arabic word. And it's one of 3 Arabic words that has existed for the longest period of time in pre-Islamic Arabic-like languages, or Northern Arabian languages. These words are luban, frankincense, he camel, she camel. And they have existed along this long period of time. It is from the language of traders that we get this word, not the language of cutters, which are the ones who would have existed down here, because we know that there was a specific terminology, sahez, for example, in the modern-day South Arabian languages. And so we don't find in the epigraphic old South Arabian languages necessarily a word for frankincense. And this can be difficult for us historically as we approach some of this material. But we know that the traders were largely descriptive in their processes when they looked at what lied before them in terms of gum resins. The word itself then comes from a Semitic root meaning white, milky white. And many Semitic words come from this base. And so we have this word in many Semitic languages and beyond. That's the power and the legacy of the word itself. Additionally, epigraphically, what is really interesting for us is we begin to find a body of incense burners, dozens of them, from Yemen, Sabaic civilization. These are written in South Arabian, epigraphic South Arabian, Sabaic, and Katabanic. On the four sides of the cube of the incense burner are what I call four notes. They are the actual gum resins or ingredients of four ingredients that can be composed together to create chords, to create perfumes. And so this kind of evidence is also important for us, but it's also not easy evidence to work from. Again, a lot of the terminology we have is descriptive. Dahab, for example, golden. So what is golden? We really don't know. We can guess which resins are gold, but we don't know specifically actually what's being used. Additionally, there's a lot of argument over one word, livnei. It appears on one incense burner. Is it luban, frankincense? Or is it storax? There's a common word in Arabic and in other languages, livni, that designates storax. So this becomes fundamentally an issue in the process. And this is also an issue when you go to source resins in the field. I've spent a lot of time now in Hargeisa and in Ethiopia. We've collected resins. Lubanja in Ethiopic and Somalia, I thought was frankincense, but it's mastic, right? And, or storax, right? And then there was another one, church, which is frankincense, right, called church. And so we really had to tease out some of the terminology. And again, you can guess it. The only way to really do this properly is chemical analysis as we build up this library that would be helpful to archaeologists and perfumers and a broader population in the region. We began then to test incense burners, and we got varying different results in the process. And this starts then the chemistry side of things, giving you all that background of the archaeological side of how this all came to be. We tried a series of different incense burners. And there's two ways to do this methodologically. One is to do it directly from the field. That's ideal. If I were to dig a site, excavate an incense burner, and then actually test it immediately. That's ideal. But we don't always have that situation. We are encouraging excavators now to test their own excavated forms as much as possible, reserving money to do this. And in our case, we're offering the services generally for free to test in the lab archaeological samples. So we've amassed probably 50 samples now. One of the particular samples we were privileged to test are a series of incense burners from the 6th century BC from the site of Ur in southern Babylonia, Iraq. And this is a major site excavated by Leonard Woolley, the British excavator who split the finds between the University of Pennsylvania, where I did my doctoral degree, the British Museum, and then in Iraq. The forms we had in the University of Pennsylvania, one particularly, we did a number of them, and we got varying results. Some we have some results, some we didn't get results at all, but we ran an FITR. That's the equipment we had in the museum at the time. And the results said something was warranted to continue testing. We saw a small peak in hydrocarbons. And we took about 4 areas of sampling here, very small amounts with a sterilized scalpel. The amounts were controlled and weighed as per the stipulations and policies of the museum. And so we are adhering to what is the standard for museums. Each museum has their own standards in terms of sampling. The process is a little destructive. I wouldn't say it's utterly destructive, but a lot of decisions were made, and there's a committee that handles this. And the questions that are sort of asked sometimes is, how much will you take? What is the weight? Where will you take? And then a lot of determination is done on where the particular object will be placed in the cabinet on exhibition. If it's low, then people would see this more, but if it's placed higher, it will be okay. So we had permission. The context of this burner is 6th century BC, the end of the Neo-Babylonian period, the beginning of the Achaemenid period, Persian period, when we know that the incense trade is well underway. And the further context is it's coming from a house. So this is the other methodological purpose that we want to define well, and that is we want to look at good context from the archaeological side. So our team, my colleague, Dr. Matt O'Connor is here, and as I talk about the chemistry, he's done a lot of our chemistry. Our team is a multidisciplinary team because it requires archaeologists, and it requires chemists, and it requires ethnobotanists and technicians to do this kind of work together. And we'll talk about the makeup of the team as we do that. As I said, the context is from a house. It's very difficult to determine whether the house collapsed into the basement in an archaeological setting, but we know that the Babylonians and Persians did bury the dead underneath the house. And so it's very likely that the context is actually mortuary. And if you think about the stench of death, the malodor of death, how something like this could be very important to mask that scent in the process. So we began to actually look at this. This is our ionized, our TIC, extracted ionized chromatograms that you see here. And we used a particular standard sample of frankincense along with the archaeological samples. And we're generating through this process some of the compounds that we're seeing. We have 3 or 4 pages of Excel compounds. And this is a good example of the Q-top chromatogram that we ran with liquid chromatography, mass spec. One of the major things that came out of this, and you can see we have the chemical features of frankincense, each bucket list, which has hundreds of features. But one of the major features that came out of using LC, and we used LC-MS because these are small molecules, and they're not volatile. So this was the reason for using LC. But the approach allowed us to be highly specific, highly focused in the process, looking specifically for key chemical features, such as boswellia acid. One of the things in this process, as we began to do this PC grouping, and Matt will be able to talk a little bit more about that. He did a lot of our PCA plotting because his specialty is spectroscopy. Our tooling showed one very interesting feature, and that is that forming a separate cluster was myrrh. And so in the process, we were able to rule out that myrrh was burned in these incense burners. That sort of evidence is very useful for an archaeologist historian. And so as we begin to talk about the frankincense burner of Oman, right, we can actually then begin to say, right, we know this is not a myrrh burner, where these burners went and how they were used. And when we talk to perfumers, for example, this makes a lot of sense. Myrrh's a lot more messy. It's a lot more sticky, right, than frankincense is. And frankincense, because of its scent, its balsam-like scent, is much more preferred in the process for good smell. So we were able to eliminate that. You can see myrrh in the PC plot on the bottom, where a lot of our archaeological samples are on the top. This better PCA2 plot, I think, shows it better. Myrrh is over here in the corner, for example. So we were using the standards of frankincense, boswellia, sacra, and commiphora as our standards in the PCA plot. The method is called MRM, and it allowed us to be particularly very targeted in the process of looking for these things as we cancel out some of the others. And we have a lot more work to do. In other words, we'll be doing one by one, taking we have hypotheses from the ancient forms that I showed you, and we have ideas of what could have been burned, and one by one we'll be targeting as we work through these again. The other thing we found was particularly 2 traces, 2 features. That was O-acetyl boswellic in the basin of that incense burner. This one came out quite nicely. We used the standard of frankincense boswellia to determine that, and our LC-MS. We had a fainter trace of alpha boswellic acid. I think you have to remember, too, that these incense burners, this one's over 100 years old. So there's a provenance history to it, too, sitting on a shelf, right, in a museum. We don't even know how it was cleaned, tossed around, removed out of a rock, brought to Philadelphia. And so we try to take as much of the information as we can about the burner in order to the provenance history of the burner, to reconstruct these things. Along the way, in order to do this kind of work, we realized there wasn't a library for us. There is a GC library, and we'll talk about that. But particularly on the LC-MS side, there wasn't a library for us to really get a good sense on the archaeological heritage side. So we began to we're building that up, right? Collecting resins, sourcing resins in the field. And the project has sort of grown now, partly because our campus is very much interested in, the UAE is interested. It's a land of perfume, just as it is here. And so we have begun the process of expanding the library into a heritage library of scents. It is all-encompassing and interdisciplinary, as I say. It is, we have chemists working with us. We have archaeologists, ethnographers. We're trying to link historical texts with images, archaeological artifacts, annotations of the chemical features and PCA plotting. The making of Bahor, that process, which you'll see in a moment. linguistic etymological data on local flora, plus then the related crafts. So really tying the tangible and intangible, these heritage features together, as well as annotated bibliographies, GPS coordinates of sourcing, and related policy issues that are associated with UNESCO and WIPO, the World Intellectual Property Organization, as they handle traditional TKs, traditional knowledge expressions, and genetic expressions. And so lastly, we hope that it'll be a library for perfume innovation in the Arabian Peninsula with the dominant data sets from Oman and the United Arab Emirates right now. Later on, we'll expand as Saudi is opening, we'll expand to Saudi and continue the work there also. And we hope to seek out collaborators in the process. It will be a password-protected database website for a while. That's to help protect intellectual property of local bahor makers. So, with permission, slowly we'll be opening things up. But it'll be a useful tool for perfumers. And I would say here too, just to highlight some of the culture of Dhofar, the lexicography work that we've been doing over the last 10 years is pretty essential in terms of documenting some of the prayers associated with Luban. So this one is Ya Luban, to you who go to the heavens, right? And so we're tagging this with the chemical feature and the material culture. Here's another one in Jabalia Shafri that talks about the use of Luban in the process. And just briefly, the last few slides then, there's an experimental side to this as we develop it. We've also then begun to work with perfume houses, fragrance houses. And we're testing old perfumes, what we think are the old creations, getting fragrance artists to make them again, and then trying them out on the market. So we have questionnaires. This is bahor making. This is actually, it's hard to read, but we've sampled hundreds and hundreds of people. We get not just the qualitative data, but the quantitative data, people telling us what perfumes smell like to them. And this is the key. You know, we talked a lot about pharmaceuticals, but there's a side of perfume complexity of chemistry here, and it's very much tied to memory, as we know, and then culture. You can see then, we've also been shooting then into this time the GC, because the library is better, and these are volatile, they're soluble. We've been shooting bahor samples, traditional mixtures into the GC, right? This is about 15 different samples from Dhofar. So we have Y-terpenine. We have particularly aromatics, a lot of aromatics from the Dhofar bahor samples, right? Just bursting with aromatics. In fact, Liaquat opened, our scientists opened them and he said, these are just smelly. And the kind of oils we're getting coumarin, which is commonly used by perfumers. We're getting Styrax, we're getting cinnamon flavors, and of course we're getting Laban. And when we compare the two, and this is the kind of work we can do now, we could take a duhun from Sharjah, which is, duhun is an elevated bahor, right? And you can see all the peaks we have, how complex it is, right? We can compare that then with a bahor from Reisut, just near here, one of the local women made, right? We can do a plus and minus of what's there, right, in the process. And this is what I hope that the library will continue to do and be a useful resource then for perfumers in the region, very much local perfumers who are learning and relearning the craft and dedicating their lives to the craft itself. It sums up our 5 steps in the process of how we do our work, ranging from the archaeological side of sampling and right through the LC and GC. And as I said, we're very much a collaborative team. Matt O'Connor, our lead scientist of the project, is here. And we work together, all of us as equals. I teach a lot of our collaborators archaeology and heritage, and they teach me chemistry. So this is a truly interdisciplinary project. And I think that's one thing you can do in the Gulf that is definitely an asset to the kind of research we do. I would like to thank our funders. The first part of the ethnography we released, the Royal Court did back in 2017 in both English and Arabic books, and we'll be producing some other publications along the side of Dhofari Bahor in the future year or two. So I would like to thank the Dewan of the Royal Court, NYU, the University of Pennsylvania Museum, and of course Dhofar University and Dr. Rashan. Thank you.
Thank you, Dr. William. The forum is now open for questions.
Yeah. Thanks for a nice talk. I mean, I just wonder about the frankincense burn basilic acid contents. You know,
Yeah.
Because we did one experiment on 250 degrees centigrade, so the boswellic acid was converted into pyrolysis, aromatized. And 250. But at 1000 degrees centigrade, how does boswellic acid survive? So how much concentration?
That's a good point. So I think Matt could probably answer that since he's done a lot of the PCA.
We don't really know. All we're looking for is just a trace marker if it was there or if it was not. So as far as like the quantities, the utility, and even the decomposition products, we're going to have to dig through the data and actually figure out what exactly it's burning into. But we can only do that just with a rudimentary survey and the LCMS, and unfortunately, I mean, it's kind of counterintuitive, but in time, as we start annotating these things, the marker may not necessarily be boswellic acid that will survive the centuries in order to locate these burners.
Yeah, the timing, and this is something we are trying to take into account. We took as much history as possible on the history of where the object went, how it was stored. Oftentimes, old boxes were pine boxes in a museum, right? So we want to be careful we're not picking up traces of the features of pine, for example. So this is part of that work as well.
So the triple quad picks it up because it's extremely focused, but the LCMS maybe not so much.
Thank you. Maybe the question not for you, William, but for your colleague.
Sure.
Because in some cases, let's imagine that there is a family, so they use Luban, and then suddenly they decided to burn Bahur. And then the house collapsed, and then archaeologists recovered remains, and what you will see? Will you see the previous frankincense that was burned in this burner, or you will see the latest thing that you burned there?
From the collapse, or the last thing that was
for the collapse.
Yeah. I think that's hard to say, right?
But you can do an experiment, and I understand that you cannot keep it for hundreds of years, but at least you see.
And that would fall under a good category.
Maybe you will see both.
I
don't know. It's
that would fall in the category of experimental archaeology.
Interesting question.
I mean, we thought a lot about this because I was trained at the University of Pennsylvania with Pat McGovern and Gretchen Hall, who they were experts in wine, particularly in the archaeological, and that's how I learned to sample. We were often wondering too, taking how many sample places do you need to take in the artifact itself? Now these are small artifacts. The circumference and so forth the width is not too long. But if you had something much larger, right? What does it tell you? So we vary the sampling technique, and when a museum will allow us, we'll take as many samples that we can, right, and from as many places as we can to see if there's any variation in the process. But yeah, I do think the hypothesis is that a lot of things probably were burned in these as resins moved in antiquity up the Arabian Peninsula, and more things have sort of penetrated the Arabian Peninsula. But there have been other teams have been testing their square incense burners, and they've been getting similar results too, not myrrh. That's the important thing. We're all canceling myrrh out, which is really interesting in the process, and they've been generally getting frankincense. The other aspect to this that's also something that has to be considered is time. So when I showed you archaeological samples, a lot of them, whatever was given to us, came from a range of chronological time, 5th century BC and then 1500 BC. And those are and the 1500 BC is up in Syria, the sites, and the 500 BC is down in the southern Iraq. Two different places geographically and two places in long history of time. And so if we can isolate those features better as we move forward, it's rewriting history as archaeologists, and that's what's really key, because it's telling us how far did Arabian trade. We often think it's first millennium BC and that's it. By 800 BC things are underway. But was it earlier than that? Particularly if a group of people could make their way, traders could make their way, which I think they could, on the eastern side and make their way up the Euphrates River, which wouldn't be impossible by maritime trade. So you don't have the camel yet, but you have boats. And if that's true, then our resin's moving up. The ancient world was a world of malodors and stench, and one way to do that is, even medicinally, right, is to wipe away that stench with these. And there's loaded questions in here, more of projects within projects. So we're interested in hearing more from all of you too on what you see here, especially those who work in the pharmaceutical side of things as traditional, sort of this traditional aromatics.
Okay, we can take one more question.
Yeah.
It's okay.
Yeah. Hello.
I have a question regarding the stability and storage of the compounds.
Yeah.
There is 2 points to it. Of course,
Yeah.
if you have like century-old stuff, you have slow oxidation, resonalization, polymerization possibilities, other things. The other one is when you talk about the fragrances, which involve regularly like aldehydes or other sensitive compounds. The question is, how? I mean, we all know that the fragrances lose with time. You can, of course, store them at -80, but even that poses some in concentration, some problems. I mean, I know from the fragrance and paper industry. So how do you manage with these 2 problems? You use artificial aging, for instance, to cover processes. And
No, they're really good questions, because I think when we started, we weren't really putting the protocols together yet for storage. I mean, I was going out to the field collecting samples and bringing them back. In the case of the Dofari samples, for example, most of them sat just in bottles, sterilized bottles, for 4 years. When we released and opened them up, they still had the fragrance. These compounds are so strong aromatics, the coumarin and the cinnamon, that they were still full of flavor. You could smell that, right? But we haven't, talked too much about the storage process. But Liaquat did do some derivatization, right? So.
That's for GCMS analysis. We have a better library, the perfume library now. So you want to backtrack with that with GCMS. What I feel we can get out of this right now is it's very qualitative analysis, not necessarily quantitative. Whereas, we know what the decomposition products would be, that may be more indicative of, what would happen later. But, for what was there before, then we can probably pick up very trace amounts of, the more abundant products that are likely to decompose. My dream is to actually, strangely enough, we have an FTICR. So actually, I would like to not necessarily look at it for compounds, but I would actually want to see the oxygen uptake of the residues by FTICR. And maybe that would that might give you some age information as far as oxygen might permeate the organics. It's an idea, but I would need to do more sampling and more analysis to see if that actually tells a story or not.
And do you link up with the standard libraries like the Mass Bank library on plant natural products and so on, and contribute to that? I mean, that would make sense. You don't have to invent the wheel new. NIST or something.
NIST is
on GC. Okay,
And our GC, we just really got up and running recently. So, he loaded that on.
My concern, and we have to test this shortly, do the residues get better signal, the archaeological residues get better signal from LCMS, or do you get better residue signal from the derivatized GCMS? Because you're going to have to derivatize them in order to see anything. And I don't know the answer to that.
Thank you, Dr. Williams. Thank you so much. Let's move to our second presentation. Dr. Sergei Dubrastu. He is UNESCO Chair of Marine Biotechnology Center for Excellence in Marine Biotechnology, Sultan Qaboos University, Sultanate of Oman. And the topic of his presentation is frankincense coating for food preservation. Please welcome Dr. Sergei.
It's working? Working? Yeah. Okay. Good. Thank you. First of all, I would like to say thank you for organizers giving me a chance to come here to Dhofar and to present my talk. So you see that we are slowly deriving and moving away from pharmaceuticals and nutraceutical compounds, but we are still talking about frankincense. Today I'm going to talk about the use of frankincense coatings for food applications. Outline is pretty standard, so I will not repeat everything. So I'll give brief introduction, and then I will move to materials, methods, and results and conclusions. So first of all, food packaging. Why food need to be packed in order to protect the food, keep it longer. And coatings protect from a number of issues. such as, for example, changes in their chemical composition, desiccation, physical processes, and pathogens. We done experiments in the previous times. We just packed fruits and vegetables, for example, okra or ladyfingers, into the plastic bags with the covered with the different sheets of polymers. And this time we want to try something different. We tried eatable coatings. Therefore, we use two components. So the major component was chitosan. So chitosan is this is the structure of chitosan. Chitosan is coming from chitin. So and chitin can be found in shrimps, crabs, crustaceans, in their skeleton, in their shells, in some mollusks as well, and mushrooms. And chitosan is a linear polysaccharide, and basically there are two ways to convert chitin into chitosan. So we use the chemical method when sodium hydroxide is used, and then it's deacetylate chitin, and then we have amino groups which became actually quite active. But also there is enzymatic deacetylation as well. Chitosan is actually used in a number of different applications. We tested this as a marine antifouling agent, also for water treatment used in agriculture, but today we're going to talk about use of chitosan as in the food industry. The second component that we use for our eatable coatings is a frankincense. I will not talk much about frankincense. So just need to remind you that it comes from trees belonging to the genus Boswellia, and we use Boswellia sacra, so local frankincense. And in particular, we use frankincense essential oils, which contain a lot of different terpenes. And of course, we have boswellic acid. This is monoterpenes, pinene, which are having strong antimicrobial properties. So the idea was to combine chitosan, which already has antimicrobial properties, and enhance these with the addition of frankincense. So we want to take, our hypothesis was to make chitosan compositive food coatings with the frankincense, and in order to extend shelf life of the strawberries and prevent growth of microbes. So the objective was to make such coatings and to investigate the effect of such coatings on the physical and chemical properties of strawberries, as well as how they affect the microbial growth. We done two types of coatings. So we use chitosan coatings because we knew previously we done experiments with the chitosan. Chitosan was effective for storing vegetables and fruits. And we want to also compare the effect of chitosan with the frankincense oil. So for this, we don't want to put high concentration of frankincense because we will change the taste of the strawberries in our case. So we put 0.5 milliliters per 1 liter of chitosan solution. So again, chitosan is not really soluble in water, so you need to prepare a solution of chitosan, you need to dissolve it in acid. So we use weak acids, such as, for example, acetic acid, and then we neutralize the solution with the sodium hydroxide. So this is the outline of the coating. So this is the first step when solution was made. This is solution was neutralized for chitosan. So in case of frankincense, we had a few drops of frankincense. And then strawberries were coated with the warm solution. So again, that the color will not change. And you see that this is during drying stage. So you have strawberries with the natural color. And then the experiment was conducted in controlled temperature chambers for 8 days at 10 degrees. And you know that if you put the strawberry in for 8 days, you know what is going to happen. It will dry, became dry, and sometimes microbes start growing on these strawberries. So we have 5 experimental treatments. So we use 1% chitosan coatings, 3% chitosan coatings, and we use 1% chitosan coatings with the same concentration of frankincense, 0.05%, and 3% chitosan coatings with the frankincense. And as a control, we use no coatings at all. We also measured physical and chemical parameters for these strawberries during the experiment. So in particular, we look for color, texture, pH, moisture content, and total soluble contents. So this slide is rather busy, but I will walk you through this slide. So for color measurements, we use spectrophotometer, and particularly we use hue angle for measurements. For texture analysis, we use hardness, a special device to measure hardness of the strawberries. For pH, we just simply homogenize the strawberries and then use a pH meter to measure pH. For moisture content, this is the long procedure. So we basically take strawberries, then weight them before drying, then we dry them at constant temperature, and then after that we weigh them again. So that's the difference gives us the moisture content. And for total soluble solids, we homogenize, and then we use a refractometer to measure these parameters. For microbial analysis, we simply do serial dilution experiments, and we were looking for 2 types of microbes we use for bacteria. For that, we use a nutrient agar, and we look for yeasts. For this, we use potato dextrose agar, and then it was presented as a number of colony-forming units after 24 hours in case of bacteria and 5 days in case of yeasts. So you can see the results. So not all strawberries survived the experiment. So you see some of them became black, and then there is some microbes are already growing on them. So first is changes in terms of the color. The control, you see the color of the control samples remains more or less the same. As you can see, different colors of the bars demonstrate the progression within the time. And then you can see that almost all treatments, they have higher color hue angle compared to control. But the best results was recorded for chitosan, 3%, and frankincense coating, which was rather stable color after 2 days of the experiment. In case of pH and total soluble content, there was no huge differences between moisture content, pH, and total soluble content. In particular, soluble contents decreased in all treatments, except when we use 3% chitosan coatings and 3% chitosan coatings with the frankincense. Hardness of strawberries decreased during the 8 days of the experiment. So you can see the control. So it's decreasing over the time. For others, I can point out the chitosan 1% coatings, the hardness remains the same. For others, it was going down and upwards. But still, you see that the standard deviation is quite high. That's why the changes were not significant. the most interesting results we obtained for bacteria in strawberries and the end of the experiment. So you can see that at the beginning of the experiment, we had about 250 colony-forming units of bacteria, and it's not surprising that number of bacteria increased in the control, almost doubled at the end of the experiment. However, if we are talking about all treatments, that the number of bacteria, most of treatments, they reduce number of bacteria. And the lowest number of bacteria was recorded for chitosan and frankincense coatings, 3% chitosan plus 0.05% of frankincense. So this shows that actually the chitosan and chitosan frankincense coatings are actually good for the inhibition of bacterial growth. And I forgot to mention that we couldn't find any yeasts in our experiment. So what are the possible antibacterial mechanisms for these coatings? We know that chitosan is actually the amine groups of chitosan interact with the cell walls of bacteria, and that's why they lead to the destruction of bacteria and killing them. So probably components of frankincense, such as boswellic acids, as well as terpenes, mono and D-terpenes. They also have antimicrobial activity, and probably they are affecting the cell membranes of the bacteria. And that's how we have a combined effect of frankincense and chitosan on the bacterial growth. In conclusion, we found that all coatings, chitosan and chitosan frankincense coatings reduce the total number of bacteria in strawberries. However, they did not affect the properties of the strawberries. However, they even have more stable color properties, and then they also retain the solid content of the strawberries. Therefore, it's possible to use such tools, such coatings with the frankincense and chitosan for food preservation. At the end, I would like to say special thanks for organizers of these meetings. And this work was not done only by me. I was a supervisor. It was done by my master student, Ms. Rahma Al-Mamari. And of course, it's like in the previous talk, it's not just one department was involved. It's a group of people. So the big help came from the Department of Food Science and Nutrition from Sultan Qaboos University. Thank you, and shukran. I hope I was on time.
Thank you, Doctor. Now the forum is open for questions.
Thank you. Thank you very much for your work. This work, we had a similar work in Morocco. Mm-hmm. It was in a project, Prima project, with some colleagues from FAS University, and exactly is the same, and the approach and it is strange. But I have two questions. The first, chitosan was extracted from marine. Why did you choose strawberries to test? Okay. In your position, for me, example, I can test for fish. Yes. To be in marine condition. This is the first question. The second, I was surprised the presence of bacteria at 3 and 4 pH of 3 and 4, mm-hmm. and the absence of fungi. But this is a strange scientific result. I don't know. Normally, 3, 4, there is no bacteria. I agree, but this is what we got in terms of bacteria.
Then in terms of your first question, why we choose strawberries, because strawberries are actually a high price product, and they easily get spoiled. In our previous experiments, we use okra, but you know, okra is relatively cheap and not as pricey as strawberries. And they even without fridge, you can keep okra for about a week, and there will be not much. So we want to target those fruits that are high value and also they easily get spoiled.
Спасибо. Also, I have a comment for that. It's a good, I think, for agriculture. You can use the remain of sense of the frankincense on agriculture for fruit trees and different flower at the house. It can kill all bacteria around the tree in the soil.
Yeah,
Thanks.
I think it's a great idea to use remains of frankincense, and previous talks showed that actually the activity, and we done similar results.
I tested at my home. I tested that.
So, oh, you tested. So
yeah.
Then you have to patent your discovery. Thank you.
Yeah, thank you. Very, very nice results. But I was missing two things. First of all, of course, what will be the price increase? So how much would it cost to do that coating? And the second one is about the properties. What is about the flavor? I mean, frankincense oil has a very strong flavor or note to it, and of course here it may be acceptable, but in the world out there might be not.
Thank you. It's a good question. So regarding the price, because it was a little amount, and we dissolve chitosan. Chitosan is not really highly priced, but especially since we are using thin films, so the prices will be increased, will be negligible for high value products. Of course, if you are using something cheap, then it makes no sense. And regarding frankincense, we specifically tested with the volunteers to see if they feel the difference, and that's why we specifically use not higher than 0.05% of frankincense. So if you increase this, then it's give a strong taste of Luban, and yeah, not everybody will eat that product unless you develop a totally different product with the Luban aftertaste or something like that. Yeah.
Thank you. Any more questions, please?
Thanks for nice talk. Just short question. How much time, they keep, preservation time, for how many days? I mean,
we tried 8 days, but I don't know. We stopped the experiment because we ran out of strawberries. So in pilot study, we kept for 2 weeks, 14 days. So probably with the manipulation, you can extend this shelf life. But this experiment was done for 8 days, and we know that in the case of frankincense and chitosan coatings, they can stay longer than that. Yeah.
Thank you.
So probably up to 2 weeks. Yeah.
Any more questions, please?
Thank you, Dr. Smith,
Okay.
for the
Thank
nice
you.
presentation. Let's conclude this session, and now